we've gone over the background on replication, transcription, translation. And those are the core processes. That's what has to happen for those processes to occur. But whether or not transcription in gene expression occurs is a whole different sort of set of things to consider. And so the topic for today is regulation of gene expression. So what really determines how do we go from that recipe at the genetic level to that functional protein? And so we'll do an overview first. Oops. Topic is regulation of gene expression. And we're going to hit both prokaryotic and eukaryotic. As you can imagine, eukaryotic is going to be far more complex. There's far more complexities in eukaryotic gene expression regulation. But we can use prokaryotic regulation to really get down deep into how the process can occur in a very specific situation. First, we're going to talk about an overview, though, to introduce some of the concepts and the language involved with it. So this is common for both prokaryotic and eukaryotic. Some genes are expressed all of the time at the same rate. So no matter the time, the place, the conditions, some genes are always expressed at the same time and rate, and that is called constitutive expression. And this occurs in both prokaryotes and eukaryotes. And so this is transcription expression of a gene at the same rate all of the time. And this, we call genes that are always expressed. Well, so they tend to be genes that code for things that are super important for viability in any cell type, whether it's prokaryotic or eukaryotic. So they tend to do essential functions in the cell. The proteins they code for tend to have essential functions in the cell. So here we can see constitutive gene expression, that the gene is always being transcribed and translated to make a functional protein. We also call these, because they have essential functions within the cell, they're commonly called housekeeping genes. So we can look here. So defending against death, getting rid of toxins, involved in energy utilization. So genes that are involved in, say, the glycolysis pathway, you have to have glycolysis. You have to have ATP for the cells to work. So those sorts of genes that do essential functions for the cell to live are always expressed. They're constitutively expressed. It depends on the cell type, the organism, but you could say in a typical eukaryotic cell, maybe about 10% of those 20,000 genes are constitutively expressed. The rest of them are regulated, meaning that something is either going to turn on or turn off the expression of that gene. And so that's our topic for today. So regulated gene expression means that the genes have to be turned on or turned off. And I'm just going to use this on and off terminology because it's a nice visual. It's a nice short little word. It's really not on all the way or off all the way. We can more think about it as like upregulated expression or downregulated expression. But I'll use the common terms on and off just to kind of, just for ease of use. So expression being turned on and off. And this is the thing that controls this is by change in the cellular environment. And that change can come in a lot of different forms. It can be exposure to nutrients, it can be exposure to toxins, it can be exposure to other cells or to some sort of matrix to attach to. So there's a lot of different environmental factors that can change, and those changes can turn on or off different sets of genes. And we can think about whether genes are turned on or off in two general ways: positive and negative regulation. So the types of regulation, the general types of regulation, we'll pick the easy one first. So positive regulation, positive gene regulation is where a gene has to be turned on. And it's turned on by the binding of an activator protein to DNA region, to a DNA region. We call that region where activator proteins bind an activator binding site, or ABS. So that's a general term for the region of the DNA where an activator protein binds. Let's look at a general scheme of this. So let's look at positive regulation first. So here's gene X. In the default situation, gene X is not being transcribed. It's off. It's by virtue of the binding of an activator protein to an activator binding site on the DNA, turns on transcription. And usually this happens by recruiting in polymerase. So what makes this bind, right? So what is it, when does it know to bind or to not bind? And that is where the environment comes into play. So the environmental factors, some of these activator proteins have to be promoted to bind, and other ones have to be blocked. They would bind by default. So the environmental factors that regulate these tend to be small molecules and effector molecules. Effector molecules are small molecules, generally, so they're molecules that promote, that bind to activators, and induce or cause the activator to bind to the activator binding site. Let's see what we got. So here is an activator protein in its default state, and there's hundreds of different activator proteins, right? And so how they're regulated can vary as well. But we're taking a generic activator protein. This activator protein in its default state is not bound to the activator binding site. Here it's saying promoter, but it would be an activator binding site near a promoter. So in the default, it's off. But by the binding of this small molecule, this could be a nutrient, it could be a toxin, it could be something that changes in the environment. When that changes and becomes present inside the cell at a sufficient concentration, it binds to the activator protein, causing a shape or a conformational change, and causes it to bind to the DNA, promoting transcription. That is called an effector protein. So an effector protein essentially positively regulates this positive activator protein and promotes transcription. Some activator proteins bind to the DNA by default. Remember, there's hundreds of different ones. So some actually bind by default. And so they can be blocked by other environmental factors, which we would call inhibitors. So inhibitors are small molecules that bind to and block activator binding. So by blocking activator binding, you block transcription. So effectors promote transcription. Effectors promote transcription by promoting activator binding. Inhibitors block transcription by blocking activator bindings. So that is positive regulation of gene expression. These regulatory proteins, called activators, bind to regulatory regions, called activator binding sites, and they promote transcription. Now, this next one, it gets a little tricky. It's negative regulation. So negative regulation of transcription occurs because the gene gets turned off by the binding of a negative regulator called a repressor. So it's gene transcription is turned off by the binding of a negative regulatory protein called a repressor. Two DNA regions in eukaryotes, those regions are called silencers. In prokaryotes, they're called operators. So negative repressor proteins promote negative regulation. They block transcription. So here's a depiction of negative regulation. When this repressor protein is not bound, when it's not bound to the DNA, to the repressor binding site, there is no transcription. When it binds to the repressor binding site, I just messed that up. Hold on, I wish I could rewind this. Let's take a pause here. Let me erase this stuff. Let me start that over and say, so when the repressor protein, here's a repressor protein, is not bound to the repressor binding site, expression is on, right? So transcription is not repressed, transcription is on. So the default here in negative systems for gene expression, the default is for the gene expression to be on. But by binding the repressor protein to the repressor binding site, either the operator or the silencer, it promotes, it shuts off transcription. It shuts off transcription. Sorry for the confusion on that. So now what causes this to bind or not to bind? So this is where the environmental factors come into play again, because some repressor proteins can be induced to bind to the DNA and other ones can be popped off. So let's see what we have here. Here's a repressor protein. So the molecules that can impact repressor binding or not, one type is an inducer molecule. Inducers are small molecules that are going to change with environmental conditions. And when they bind to the repressor, they cause a shape change. Let me go ahead and introduce that language because I don't think we've used it yet officially. They cause a shape or a conformational. Another term that can be used is an allosteric. So they cause a shape, conformational, allosteric change in the protein in the repressor that causes it to dissociate. Okay, let's start from scratch here. Here we've got a repressor protein. It's binding to an operator in a prokaryotic system. There's a DNA binding region on here. So this region recognizes the operator sequence, causes the repressor protein to bind. In this situation, the default for this repressor protein is to bind to the operator and block transcription. An inducer molecule is a small molecule that's going to change with environmental conditions, bind to the repressor, And look at that DNA binding region. It's caused an allosteric conformational shape change in this repressor protein, which causes it to dissociate from the DNA. And remember, this is a negative regulator. So when it dissociates, this is going to induce transcription. So ultimately, inducers induce or cause transcription. So some repressor proteins are controlled by inducer molecules. Other repressor proteins, well, I don't think I have an image for it here, but we'll go ahead and other repressor proteins can be negatively, well, I don't know how I want to say it, can be regulated by co-repressors. So co-repressors are small molecules that bind to the repressor, and they actually promote repressor binding. Therefore, co-repressors promote turning transcription off. So if we use this system here, if we look at this situation, say this repressor is actually controlled by a co-repressor type of regulation. So in the default, it would hang out in the cytosol and not be bound to the DNA. But in the presence of some small molecule, a co-repressor, it would bind to the repressor and cause it to bind to the DNA, therefore shutting down transcription. So those are some general sort of schemes for regulation of transcription.